Okay, so now we're going to talk about some of the organelles. So the nucleus, ribosomes, endomembrane system, and some energy-related organelles. So when we're talking about the nucleus, it's really the cell's command center, and it's the most important organelle if we're talking about a eukaryotic cell. So inside the nucleus, you can find some chromatin, which is really just uncoiled DNA that's mixed with proteins and a little bit of RNA. It kind of looks like spaghetti underneath a microscope. But when a cell is getting ready to divide, this chromatin is going to coil up tightly to form chromosomes. So that's that classic X-shaped structure that you've probably seen in biology diagrams before. DNA is going to be organized into genes, and the genes are what are actually holding the instructions for building a specific protein, or at least part of one. DNA never leaves the nucleus. Instead, the instructions are going to be transcribed into messenger RNA, or mRNA, which then is going to leave the nucleus, go to ribosomes to get translated into a protein. There's also a nucleolus, so this is a darker region inside of the nucleus, and that's where ribosomal RNA, or rRNA, is made, and it's going to be combined with some proteins to form ribosome parts. All of this is wrapped in a nuclear envelope, which is a double membrane with nuclear pores that allows some molecules in and out. So it allows like mRNA to leave the nucleus and it allows proteins to come in. So ribosomes are really the site of protein synthesis. So sometimes they're referred to as protein factories. And their function is really to read the instructions from the mRNA and build proteins by linking together amino acids. So you can think of them as like the assembly lines in a manufacturing plant. So ribosomes are found in both prokaryotic and eukaryotic cells, and this kind of tells you how ancient and essential they are. Structurally, they're made up of two subunits consisting of RNA and proteins, and they basically clamp onto the mRNA and start assembling the protein itself. So in eukaryotes, you can find them in two places. They can either be freely floating around in the cytoplasm. If they're free floating, they're going to make proteins that stay inside of the cell. You can also find them attached to the endoplasmic reticulum, at which point we call it rough ER, and that's where they're making proteins that are either going to be shipped out or inserted directly into the membranes themselves. So now we can talk about the endomembrane system, and this is going to include the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, plus a bunch of vesicles. This system is really about compartmentalization, so helping make sure that we keep different cellular processes in different areas. That way everything runs smoothly. So the endoplasmic reticulum is really just a network of flattened sacs and tubules, and it comes in two different forms. We can have the rough ER, which I just mentioned previously, or the smooth ER. So rough ER is called the rough ER because it looks rough because it's studded and covered with ribosomes. So as proteins are being made, they're going to be modified in the ER lumen, which is just the inside space. Then they're going to be packed into transport vesicles and headed to the Golgi apparatus. Smooth ER is smooth ER because it doesn't have these ribosomes studying it, and it's going to be the site of lipid synthesis. So things like phospholipids, steroids, and even detox enzymes. So in liver cells, the main function really is detox of drugs and alcohol. They would have a higher proportion of smooth ER because of this. So once our endoplasmic reticulum has done its part, it ships the products to the Golgi apparatus, which is a stack of membranes, and it kind of acts like a mail sorting facility. So it's going to receive these vesicles, it's going to modify the contents, repackage them, and then ship them to their final destinations. Some are going to be sent outside of the cell, some are going to be sent to the membrane, and some are actually going to become lysosomes. So lysosomes are really the cell's recycling and digestion centers because these vesicles are full of digestive enzymes. And these enzymes help break down worn out cell parts or even foreign invaders like microorganisms. An important side note, sometimes our lysosomes don't work correctly and things can go wrong. So one of these examples is Tay-Sachs disease, where they actually have a missing enzyme, and that's going to cause the buildup of lipids inside of the brain. And then we have vacuoles. So these are just larger versions of vesicles, mostly going to be found in plant cells, and they have a wide range of jobs. So if we're talking about a plant, they can act to store water. They store pigments like those responsible for flower color. They can even store toxins for defense. In animals, we have special vacuoles like adipocytes, and their function really is to store fat. And some single-celled organisms can actually use vacuoles to help pump out excess water or help with digestion. Okay, so now we can talk about the power plants of the cell. So that can be our mitochondria or our chloroplast. And these organelles are really going to handle energy production. And interestingly, they each have their own DNA and ribosomes. So this really supports the theory that they were once independent bacteria. So mitochondria are found in both plant and animal cells, and they perform cellular respiration. And cellular respiration is the process of breaking down glucose and oxygen in order to make ATP, which is our cell's energy currency. They're surrounded by a double membrane. The inner membrane is going to fold into cristae, and the whole purpose of these folds is really to increase surface area. Inside the inner membrane is the matrix, and this is where enzymes are going to help break down carbohydrates. It's basically the engine room, so it's where we're generating ATP so the cell can do things like move and divide and build different macromolecules. Just an interesting fact, your mitochondria is only inherited from your mother, so you don't have any mitochondrial DNA from your father. So chloroplasts are going to be found in plants and algae, and they kind of do the opposite of mitochondria. So they use sunlight and carbon dioxide and water in order to make glucose, and the process of doing this is called photosynthesis. Basically, it's solar-powered sugar production. Chloroplasts are going to have a double membrane as well, and they have an internal fluid that's referred to as stroma, and they have stacks of thylakoids, which are like little green pancakes, and they contain pigments such as chlorophyll, which are going to help capture the solar energy. So just like mitochondria, they have their own DNA and ribosomes. Next we'll talk about the cytoskeleton and cell movement.